Hi, my name is Lauren Graham and I'm a rising senior at Catalina Foothills High School.
My name is Priti Kari and I'm a rising junior at Basis Tucson North.
My name is Jorge Ruiz and I'm a rising sophomore at the University of Arizona. This summer we worked in Dr. Kilo's lab, which focuses on gastroenterology and nutrition.
Our lab is looking into inflammatory bowel disease, which is an umbrella term for conditions where the GI tract is chronically inflamed. We're interested in IBD because over 6 million people suffer from the condition worldwide and there is currently no cure.
Symptoms can be severe and debilitating and can threaten the possibility of death. Our lab's main research question is, what is the cause of IBD and what are some possible therapeutics?
During my time in the lab, I looked into the role of retinoic acid in PARP1 in DSS-induced colitis. The inhibition of PARP1, which is an important enzyme in DNA repair and transcription, showed decreased colitis severity and increased LDH1 enzyme expression. Now, LDH1 enzyme expression is significant because it plays a role in retinoic acid synthesis, which in turn plays a role in controlling inflammation in the body. While I was in the lab this summer, data concluded that LDH1 gene does have an effect on these mice, as mice without the gene had increased colitis severity. As the summer comes to the end, the lab hopes to explore the combined effect of PARP1 inhibition and the deletion of LDH1 gene on DSS-induced colitis in mice. This is all in the hopes to conclude whether the deletion of the gene masks the protective effect offered by the inhibition of PARP1.
My project is exciting because it is at the forefront of research into the gut microbiome, which is an area of interest for many different medical fields. Specifically, I have been looking into a relationship between fecal metabolites, which are part of the gut microbiome, and a cellular process called phosphorylation. Previous data from this lab has shown that fecal metabolites regulate the amount of phosphorylation occurring. We are interested in this because high levels of phosphorylation have been related to chronic inflammation and IBD. If we can find out which fecal metabolites are causing increased phosphorylation, we might be able to inhibit those metabolites and alleviate symptoms of chronic inflammation. Additionally, part of my research question is to look into which proteins are being pegylated. This will give us greater insights into why high levels of phosphorylation are related to inflammation. The project that I'm most closely involved with is the proteomic analysis to determine which proteins are being pegylated. While we didn't complete it with the given time, I was able to contribute to experiments that gave us data that will be used in the future proteomic analysis.
This summer, I worked on a project that focused on the downregulation of a protein called disabled homolog 2, or DAB2 for short. When DAB2 is downregulated, it causes a pro-inflammatory switch from a tolerogenic switch in many dendritic cells. TLR4, on the other hand, is a toll-like receptor that causes signaling pathways to induce immune and inflammatory genes. This pathway is activated by LPS, which downstreams the activation of AKT kinase. This lab has found in the past that AKT kinase phosphorylates DAB2 at 6 possible sites on the surface of DAB2, but we aren't sure which of these sites are responsible for its downregulation. Using Western blotting, we ran samples of transfected cells with mutated DAB2 to see downregulation patterns of the protein. We found that sites serine 227 and serine 394 on DAB2 are responsible for its downregulation by phospho AKT.
I'm curious about how we can more accurately create models of IBD in mice and in vitro. I also want to know more about if there are specific fecal metabolites that are responsible for hyperpyrrolation in IBD patients.
This summer, I was curious about how we can use machinery to replace manual work that we do in research, and to what extent is this more or less efficient to further scientific research.
And I'm curious about the combined effect about new mice that Dr. Kilo will be creating in his lab that have PARP1 inhibited and then a deleted LDH1 gene. I want to see which one has a stronger effect on the severity of colitis symptoms and if the deletion of the LDH1 gene masks the protective effect of the PARP1 inhibition.